You're now at the lab with the Cold Spring Harbor Laboratory. I'm Sam Diamond, and this week at the lab, the golden grail of genomics. The human reference genome is one of the greatest achievements of the modern scientific era. It pushed biology into the 21st century and revolutionized our understanding of genetics. But, at the end of the day, it's just a reference. One genomic profile standing in for all of us. In reality, each of our genomes contains about 4.5 million genetic variants. Today, thanks in part to scientists who are involved in the Human Genome Project, it's no longer cost prohibitive to sequence any given individual's genome. One such scientist is CSHL professor Thomas Gingeris. After the Human Genome Project wrapped up, Gingeris became involved in a follow-up project called EN-CODE. Short for the Encyclopedia of DNA Elements, this genomic database opened a world of possibility. Gingeris was quick to dive in with EN-TEx.
This project started at the end of phase 3 of EN-CODE. It was clear that more and more individual genome sequences were going to be used in place of the reference human genome sequence. And the question then becomes, what are the differences that you would see using the reference genome versus taking individual genome sequences and then doing the same kind of analysis?
If EN-CODE was the sequel to the Human Genome Project, then EN-TEx is the third installment in the trilogy. Gingeris launched EN-TEx alongside Yale professor Mark Gerstein. Together, they led a team in profiling 30 tissues from 4 donors with 24 different genomic tests per tissue. As a result, they were able to compile a catalog of more than 1 million genomic variants, making it the most comprehensive per-genome resource of its kind. But they didn't stop there. They also developed computational prediction models for evaluating each individual genetic variant in cases where collecting a tissue sample would be overly invasive, like if you're looking into heart or brain conditions. How does Gingeris feel about all this?
I think it's quite exciting. I mean, this is the golden grail that people have been wanting to do for a long time. And this is really the first attempt to provide a paradigm for getting to that goal.
And it started right here at the lab. Thanks again for joining us. If you like what you heard, please subscribe wherever you get your podcasts and visit us online at cshl.edu. For Cold Spring Harbor Laboratory, I'm Sam Diamond, and I'll see you next time at the lab.